Hello students, welcome back to a new video of Sunanda's tutorial. In this video, we will discuss double fertilization in angiosperms. In my previous video, I have discussed about the pollination. So today, after the pollination, the next steps we will discuss here. The next steps after the pollination. So what is fertilization? It is unique to angiosperm and called as double fertilization because each pollen grain after germination produce two male gametes. One fuse with your egg, other one fuse with your polar nuclei. So as two fertilization occur togetherly, it is called as double fertilization. One of the sperm fertilizes the egg cell to form zygote, while the other fertilizes the two polar nuclei, resulting in the formation of endosperm cell. Okay. Next, the phenomenon of fertilization was first reported by Strasburger in 1884 in the plant Monotrappa. The male gametes are brought to the egg present in male gametophyte by the pollen tube. So angiosperms form pollen tube, and through that pollen tube, pollen comes near the egg. Then this phenomenon is called siphonogamy, which was discovered by G.B. Amici in Protulaca plant. Okay. Next, steps of fertilization. Different steps include: after pollination, the pollen grain germinate on the stigma. Then the pollen grain move through style. Then it enters into the embryo sac. Then it discharge the male gametes inside that embryo sac. Then fusion of gametes and double fertilization. Next, the male reproductive organ of flower is called as stamen. It produces the pollen grains. Okay. Then the opening of anther or dehiscence of the anther makes pollen available for pollination. After anther dehiscence, pollination will happen. Each pollen grain contains a vegetative cell and a generative cell. Generative cell divides to form two sperm cells or male gametes. Pollination takes place through abiotic factor or biotic factor. Once pollen grains settle on a compatible pistil, it may germinate in response to a sugary fluid secreted by mature stigma. Lipid at the surface of stigma may also stimulate pollen tube growth for compatible pollen. Okay. Next, germination of pollen. Then at first the stigma will recognize that pollen grain. Only compatible pollen grain after reaching the stigma will recognized by the stigma. Then it will germinate. So at first recognition, pollen grain reach the receptive stigma of the carpel by the act of pollination. The female sporophyte must recognize the pollen stuck to the stigma. Then only the pollen of same species can successfully grow, that means successfully germinate. Then initiation. Once the pollen grain is recognized and hydrated, the pollen grain germinates to grow a pollen tube. In pollen grain, the generative cell gives rise to sperm, whereas the vegetative cells have a tube cell that grows the pollen tube. Next, growth and signaling. The pollen tube growth is influenced by interaction between stigma, style and pollen grain. The elongation of tube is achieved with the elongation of cytoskeleton, and it extends from tip, which is regulated by high level of calcium. So the role of calcium is very vital here. The calcium levels help the synaptic vesicle in the membranes grow and extend at the tip. calcium ion plays important role in pollen germination. It is a very important point that calcium ion plays important role in pollen germination. Then polypeptides found in style also regulate the growth of the tube, and specific peptides that play a role in signaling for growth have been identified. Emerging data indicated that two synergids on the side of egg cell emit a diffusible species specific signal to attract the pollen tube at the last step of pollen guidance. Cysteine rich polypeptides in a subgroup of defensin like proteins are attractants derived from synergid cell. So synergids release some kind of chemicals which will attract the pollen tube inside the embryo sac. Then in some species, lure peptides that are secreted from synergids attract the pollen tubes. Other parts of the ovary include cytoplasmic factor like miRNA and other chemical gradient that attract the pollen tube. So in every species there is a specific chemical which will attract the pollen tube, and that chemical is secreted by the synergids. Next, so it is the whole scenario, whole picture of your interaction between stigma and pollen tube. Next, the elongation of pollen tube through style. Next, during germination pollen grain depends on many factors. One is the presence of high calcium ion. Next, by absorbing water they swell and gets activated. Okay, the other factors that are required are carbohydrate, calcium, boron. Apart from calcium, boron plays major role during this pollen germination. Some enzymes also plays important role, some hormones also plays important role. Optimum temperature ranges between 20 to 30 degrees Celsius for pollen germination. During germination, pollen tube, all content of pollen grain migrate to the pollen tube, and whole growth is confined to its tip. That means the growth of the pollen tube occur from its tip. For the confinement of the cytoplasm in the tip of the developing pollen tube, a number of callose plugs are formed. Okay, so many callose plugs are formed which helps for the confinement of the growth of that tip only. Okay, then pollen tube walls of flowering plant contains callose that resides in the tube wall and forms the plug that separates the growing tip from the evacuated tail. The callose plug keeps the pollen cytosol within a reasonably small volume and therefore has been thought to play important role in fertilization. Then path of the pollen tube. After the emergence of pollen tube from pollen grain, it makes its own way between stigmatic papillae. So after the germination, pollen tube is formed. Then pollen tube will move through this stigmatic papillae and then the style. Histologically, styles are of three types. One is open type, where transmitting tissues are dissolved by pectinase enzyme. Some are half closed. Stylar canal remains surrounded by rudimentary transmitting tissue, and some are totally closed. There is no open channel but a solid core of tissue. So there are many types of styles are present. But anyway, pollen tube make its way through that style and finally reach to the embryo sac. After reaching near the embryo sac, it will either enter through micropylar end, which is called as sporogamy, and This sporogamy method is guided by the filiform apparatus which is present in synergids. Then chalazogamy or basegamy, where pollen tube enters the ovule from chalazaland, and this entry of embryo sac occur through however the entry of embryo sac occur through micropyle, but the final entry occurred through micropyle. Then mesogamy, where the pollen tube enters the ovule through funicles or integuments. But in each and every case, the final entry inside the embryo sac is through micropylar end, because on that micropylar end one hole is present. So it helps the entry of the pollen tube inside the embryo sac. Then obturator, it is a special structure swelling of placenta near the micropyle of each ovule, which helps or facilitates the entry of pollen tube into the ovule by forming a sort of bridge. Soon after fertilization, the obturator shrinks and disappears because its role is over. So what we have learned from this slide, we have three pathway for the entry of the pollen tube inside the ovule, but finally it will enter the embryo sac through micropylar end. Then we have a special kind of structure called obturator, which facilitates the entry of pollen tube inside the ovule by forming a sort of bridge, and this structure will disappear after the fertilization. Okay, next, so these are three pathway for entry of pollen tube inside the ovule. Then entry of pollen tube into embryo sac. Now, irrespective of place of entry of pollen tube into ovule, the pollen tube always enters into embryo sac through micropylar end. It is a very, very important thing to remember. Then this may be different types. The pollen tube may be enter between egg and synergids. It can enter the embryo sac wall or synergids directly through synergids. Then discharge of male gametes. Inside that pollen tube, male gametes are present. The pollen tube contains two sperms or male gametes or haploid male gametes. When pollen tube enters the embryo sac, it bursts and releases the content, two sperms along with certain amount of protoplasm. And the first male gamete discharge in one of the synergid. The pollen tube successfully burst. They always discharge into degenerated synergids. In addition to this pollen tube dependent promotion of synergid degeneration. So synergids plasma membrane, one of the synergids will be destroyed after the release of one sperm inside it. Next, fusion of gamete and double fertilization. So one of the male gamete will fuse with the egg and other with the polar nuclei. So inside that embryo sac we have 8 nucleus. 3 are arranged at the top, 3 at the bottom and 2 in the middle. 3 antipodal cell, 2 polar central cell, 2 synergids and 1 egg cell. One sperm or male gamete fertilizes the egg cell and form the zygote. So one male gamete will fuse with the egg cell to form zygote, which is diploid in nature. Okay. Another male gamete will fuse with the polar nuclei to form a triploid nucleus, because polar nuclei are 2 nucleus, one large cell having 2 nucleus. So one male gamete, another male gamete will fuse with that polar nuclei to form triploid nucleus, and which is called as primary endosperm nucleus, PEN. So one male gamete will fuse with egg to form the zygote, another male gamete will fuse with the polar nuclei to form primary endosperm nucleus, which is 3N, that means triploid in nature. So this double fertilization was first observed by Novastine in 1898 in Fritillaria and Lilium, and this method, which was discovered by Novastine, was supported by Gignard. So the fertilization technique was discovered by Strasburger. Then double fertilization was discovered by Novastine, and it was supported by Gignard. Okay, next, it results in the formation of triploid endosperm nucleus, that means the fusion of polar nuclei and a male gamete. Then endosperm is therefore triploid in angiosperm, and it is a specific characteristic feature of your angiosperm. Endosperm serves to provide nutrition for developing embryo. So after fertilization we have two things, one is zygote, another thing is endosperm. So that zygote will develop to form embryo, and for that development and growth the nutrition is provided by the endosperm. Okay, next, it is the figures of double fertilization, and it is the whole life cycle of your angiosperm. So male gamete and female gamete in this way fused with each other to form zygote, and zygote is the first structure of the next generation, and it will develop to form embryo. From embryo then seed will be formed, and from seed the next plant will be can be developed. So it is the whole life cycle of a angiospermic plant. So it is all about your double fertilization and triple fusion. In my next class I will discuss how embryo is developed in dicot and monocot plant. So I will discuss post fertilization events in my next class, and then your whole series of this sexual reproduction in angiospermic plant will over. And please stay tuned, and if you liked it, please hit the like button, please share with your other friends and family members as well as teachers, and please subscribe to my channel Sunanda's Tutorial. Thank you.